Okay, may I know you are doing your PhD or masters? Or your class is actually a mix of masters and PhD? Okay, so all of you is a mixed mode master program. Okay. I mean, in that class right now, do you have microphone that you can interact with me? Can answer my question rather than using chat box? Okay. All right. So in the classroom right now, there are only two students. So another student is actually at their own place, right? I mean, they are actually online. Is that the case? Okay. Okay. So shall we start now? Can we start the seminar now? Okay. Because I'm going to close my chat box. So that's why I actually asked you all whether you can communicate with me. Okay. The audio communication. Okay, so just now that you just had the seminar with Prof. Ada, she talked about all the um, regulation and the requirement and ethical issues in doing research using animal. Okay, so I'm going to continue a little bit on that, talking about animal experimentation and the principle of animal care and welfare. These are all actually related. Mm. Yeah. Okay, so I'm going to talk a little bit on the, of course, benefit of animal experimentation, and we go through the importance of animal model, why you need to use animal model in your in the biomedical research, and what type of animal model is available, or category of animal model, and at the end we are going to look at the principle of animal care and welfare. Right. So my question is, how many of you uh, plan to conduct animal experimentation? Okay, I'm open my chat box. You can none of you. Okay. So you do not have in your masters. You are not going to conduct any animal experimentation. Yes or no?
No.
No. Okay. No one. But of course, when you go through the all the experiment type of experiment, you may have some ideas or maybe thought about these animal experimentation. So, what is your stand? I mean, are you actually support animal experimentation or you're against animal experimentation? Can anyone who actually against animal experimentation tell me something? Is there anyone from here? No? Anyone to speak up? This is because it's quite interesting to share your view if you are against animal experimentation. So myself also would like to know what is your point. Okay, because of course there are arguments about the benefit and the problem of animal experimentation. Okay, and then of course I support animal experimentation. Then it's just like because they actually have a lot of benefits. Okay, so in the area of when you study autoimmune deficiency syndrome, behavioral, cardiovascular disease, cholera, hemophilia, malaria, and so on, I put it here. Okay, it's all give some benefits. Okay, we know that it's actually benefited human and non-human health, and these things is actually achieved by using animal model. All right, so as I said that um, lots of animal involved. Okay, then I just want to highlight that without animal experimentation, polio would kill thousands of unvaccinated people each year. I know that now with the COVID-19, you have mRNA vaccine that you don't have to, involve animal. Okay, but now a lot of vaccine is still produced in animal. All right, so people disabled by stroke or spinal cord injury could not benefit from rehabilitation technique. Some of the rehabilitation techniques is actually available because of animal experimentation. All right, so also chemotherapy as well. So why uh, we can actually use animal or the advantage of using animal. Okay, number one is laboratory animal have organ and body system similar to human. Okay, if that they are not similar, then we cannot use animal. So we use animal because their organs and body system similar. Because they are similar, that means we can study human system, condition of human organs in animal. So whatever, of course, result some result is not really direct. We have to carefully interpret the result what we got from animal experimentation. But in general, because they are similar, they have similar organs, heart, kidney, uh, uh, gastrointestinal, and so on, brain, whatever. So then we can use these animals. This is the reason why we can use animal experimentation, okay, to study human. All right. So number two, in many cases, they are susceptible to the same disease that affect humans. Okay, not only they are having similar organ and body systems, okay, they also contracted or susceptible to the same disease as us. So we can study also in animal, all right, to understand maybe the how disease occur and to challenge them to find some cure as well, right? Next is actually the short lifespan of animals. Of course, you want to study some type of experiment. You want to study for the the lifespan of animal, so you don't have to wait years and years. All right, because lifespan of the animal is shorter, so you can study and repeat the experimentation. Okay, if you study in human, then you have to wait, 60 years or 80 years to complete your study. So that this is not possible. All right. Number four, laboratory animal in scientific study allow researcher precise control over their environment. So you can control their environment, not only their environment, actually, you can actually create a certain animal with disease, like, or even like, with obese, or you can create animal with hypertension, okay, and you can control a lot of things. So in human, it's actually is not possible. Okay, you also can control the environment that they actually live and maintain as well. Okay. So experimentation is often not possible or not ethical in human subjects. There's a lot of things that's not ethical to be conducted in human. So this is the issue. Some people say, "Oh, why you use animal? Is that if not ethical in human, then is it ethical in animal?" Okay, so this is the argument for people who actually against animal experimentation. Okay. But that's the fact. So type of animal experimentation. So I just now is actually why we use animal. So now is the type of animal. It depend on what things that you want to study. If you use the animal model to study the biological and functional system in human, okay, to study the biological and functional system in human, normally you divide the category in these three category. Number one, exploratory animal model. So exploratory is animal used to gain an understanding of fundamental biological mechanism, whether the mechanism operative in fundamental normal biology or mechanism associated with the abnormal biological function. So this is actually, they put all under this category. So you want to explore some fundamental mechanism, whatever. So that's why they call it exploratory animal model. So any animal model, if you use them to explore, to understand the fundamental biological mechanism, then this is actually fall under this category, exploratory animal model. So if you want to use the animal to gain an understanding of complex biological problems, it's actually under explanatory animal model. All right. And when you use the animal to discover and quantify the impact of investigative treatments, okay, like the one that I just said, that you create mice with hypertension or hypertensive mice. Then after that, you want to challenge with some product, some maybe drug or some herbs product, whether it actually can reduce the blood pressure, whatever. That means you want to study, you want to investigate whether that compound can be used as a treatment. So this is actually you use the predictive animal model. Okay, so remember, when you use for the biological and functional system in human, this all fall under these three categories. But if you use the animal for disease, in disease research, there's a lot of studies in USM. We have a couple of researchers from Inform as well, okay, and other school of pharmacy, and in health campus, a lot of studies using animal in health campus and IPPT as well. Majority of the study conducted in USM is actually for the disease research. Okay, so for this, there's a lot more category. Okay, because this is what they are doing. Number one is actually induced animal model. All right, the one that I just said, the obesity, the obese mice, or hypertensive mice, or even diabetic. Okay, you can use certain chemical to induce the diabetes in rats. Okay, so when you conduct that type of things, it's actually induced animal model. So where normal animal, you experimentally created, or either through surgical, some genetic modification, like the hypertensive normally is actually the genetic modification. So the the strains, the one that is actually we have, generally they are hypertensive, okay, through genetic modification. All right. So for diabetics, normally achieved through chemical injection. All right. So that's the induced animal model. So the spontaneous animal model is genetic variant which mimic the human condition where the variant occur naturally. It's just like about the same in subset to number one, but it's actually occurs spontaneously. All right, then you just maintain that the animal that carry the genetic variant in them. All right. Number 3, transgenic animal model. So this is quite famous. You create the transgenic animal model. This is actually under two category. So this is induced model in which DNA inserted. Well, just like when the word transgenic normally is just like refer when you insert the DNA. So what is the purpose of inserting DNA? Because you want to express the type of protein that you want, okay? Or you want to express the same type of protein, but in a the increased amount. You want to duplicate or triplicate the amount of the original expression of that protein. So normally you create the transgenic animal model. Okay, when you want to multiply, when you want to increase the level of expression of whatever protein that you want, or totally different protein you want to express into the cell or animal. This is transgenic. But when you stop the expression of the normal gene, okay, because our cells have got expression of different type of gene, but you identify one or two genes, or normally just one gene, okay, because one gene, then you stop the expression of that gene. There's a lot of techniques to achieve that. That condition, we call it knockout. Okay, you knock out. So it's a knockout model. All right. If you knock out two type of genes, normally we consider as a double knockout. All right. So you have to. This is actually two category. They are different. It's just like in opposite to each other. All right. When you create transgenic, it's actually you increase the protein expression. When you produce the knockout, you stop the protein expression. So like when you stop the certain protein expression, then you can see if the protein is actually important for the eye development. If you stop the expression during inside the embryo, then you will not see the eye develop normally. This is what the purpose of that. So negative animal model is actually when the animal fail to react to a disease of chemical stimulus. Sometimes you do the study, you want to develop the disease or the model, you put some chemical, whatever, but they fail to, to react. This one, you have to reevaluate that. It maybe is very valuable because you can use them as a negative animal model. It's just like a lot of things in experimentation. When you do experiments, sometimes things that's not working very well or turn out to be negative for you is actually a big thing. Like when researchers discover penicillin, right? So they actually in the lab, they keep growing the bacteria, but they found out that the bacteria is actually fail to grow, but then they find out that this is actually something killing them. It's actually they discover the penicillin, right? So this is something that quite famous story. All right. So number 5 is often animal model, where animal in which a disease occur, but there is no corresponding in the disease in human. The disease is actually specifically occur inside the animal, but still can give us some insight into our understanding of that type of disease. All right. So for animal welfare, so just now Prof. Aida talked about the regulation or the act govern our using of our use of animal in experimentation, and the things that is actually discussed inside the regulation or the act is actually how you treat the animal, how you take care of their welfare. Okay, because there are good ethical, scientific, and legal and economic reason for making sure the animal are looked after properly. All right, because good science and good animal welfare go hand in hand. So you just imagine, because you want to have your animal healthy, you want to keep your animal healthy. When you do the experiment, the result that you generate from your animal experimentation will be valid. Okay, if your animal is not healthy, they are sick. Sometimes that when you have your result, it's not actually coming from your experimentation. It's because the animal is already sick. Okay, like the animal that you grow, that you hold the animal, or you just like raise the animal, then animal is actually carry something, like maybe they have already contracted certain disease inside their liver. Then you study the liver, you see something abnormal in the liver, but it's actually not coming from your experimentation, because they already contracted some disease, some liver disease already. So that's why we need to make sure that the animal is healthy. Okay, and then it's going to affect your result as well. Okay, and then you have to imagine that the animal used for research, okay, is actually kept within an animal facility. All right, they are totally dependent on the researcher, on the staff, on whoever assigned to look after them. Okay, so uh, researcher and student is actually need to give attention and take care of their health and well-being. All right, so that's why you need to inspect animal at least once daily, and perform routine examination um, to to detect any abnormalities, or they are not feeling well. So you need to know what parameters that you need to look after. Yeah. Then after you discover the animal, normally the animal is quite easy. Okay, when you do animal experimentation, it's quite easy for you to monitor the animal well-being by looking at number one is animal when they sick. I think it's just like the same to human. Okay, we just speak out when you are not feeling very well. You speak to people. You tell people that you are not feeling very well. But for animal, they cannot talk to you. But the things that when you are sick, you stop eating, you stop drinking. Okay, so when you put your feeding things or the food to your animal, and you discover that the next morning the animal is not eating, all right, then it's something wrong with the animal. Okay, and you can also look at the how they groom themselves, because animal is quite it's just like just like uh, we, okay, when we've got some issues, so they stop grooming ourselves. So when you go to see your friend can detect that. Normally they say, oh, what's wrong with you? You looks like, a bit messy, a bit not very well. So like animal, they stop grooming, okay, themselves. So you know that your animal is not having some problems. Then you have to let the vet surgeon or the vet officer know about this, and they can come and have a look at the animal and identify what's this problem with the animal. So you can actually uh, uh, give some medicine, or decide from that. Okay. So there are principle. This actually developed the US government principle for the utilization and care of vertebrate animal use in testing, research, and training. Okay, just like just now, Prof. Aida already mentioned to you, when you use the animal model, and whatever govern is actually they use animal for research, testing, training, and teaching. Okay, the same thing. So training and teaching is under the same category. All right. So this is the 9 US government principle. They are foundation for human care and use of lab animal in US. It's actually been adopted by the rest of the world. Okay. So this principle were developed by the Interagency Research Animal Committee and adopted in the US as part of their policy on human experimentation. And it's actually the same principle used under the ICOOP as well. Okay. So we just quickly go through this principle. So far, do you have any questions so far? Right? If not, then just continue. Number one is actually a principle on the transportation, okay, care and use of animal. All right. So transportation, care and use of animal should be in accordance with the Act. If you have Act, like in Malaysia, we have Animal Welfare Act 2015, okay, and other applicable law, guidelines, and policies. So, in USM, as Prof. Aida mentioned, that we have ICOP, okay, the ethical committee of the ethical committee in USM, and we have certain guidelines for you when you do the how you transport the animal, how you take care of the animal, how you use the animal. Okay, so this is actually in place already. All right. So this principle said that we have to have this in place, and not only at the institutional level, but we should have this at the national level. As a country, part of the scientific community, that we have to have the Animal Welfare Act. So we are actually one of the country that our parliament actually approved and mandated this Animal Welfare Act in place. Okay, for us to, it's actually to govern the use of animal. All right. Number two, procedures involving animal should be designed and performed with due consideration of their relevance to human or animal health, the advancement of knowledge, and the good of society. So just now we talked about the uh, the animal model. Okay, the type of animal models. So you need to actually select the correct animal model for you to be used. All right, because you want to have the relevance to human. Of course, I said that why we use animal, because animals share a lot of things similar to us in terms of their organ system, but still there are a lot of species of animal. Okay, so when you want to do some animal experimentation, you also need to consider what type. You want to use rabbit, you want to use mice, you want to use rats, because they are all have their own advantage, or in terms of similarity to human. But some experimentation you need to better perform in rabbits. Okay, but general principle is that if you have option like mice and rabbit, you can use both. Normally you use in rabbit, you use in the smaller one, because of course using smaller animal, so you actually the cost is also reduced. All right, and then the principle is that you actually sacrifice smaller animal rather than bigger animal. Okay, and of course Prof. Aida also talking about use minimum numbers of animal. Yeah, because here you can we can have the 3R principle as well. Okay, they also have under the animal welfare principle also cover the 3R principle. Okay, number three, the animal selected for a procedure should be of appropriate species. Okay, as I mentioned just now, appropriate quality, and the minimum numbers required to obtain valid result. Okay, you need to have to use minimum numbers of animal wherever applicable. If you have mathematical model, computer simulation, or in vitro biological system that you can use instead of using animal, that should be given priority. Okay, or you can use in combination. Okay, your study will greatly reduce the numbers of animal by considering these alternative methods. Okay, this is the thing that you need to consider. And of course, the proper use of animal, including the avoidance or minimization of discomfort, distress, and pain when consistent with the sound scientific practices, is actually uh, important. All right. Unless the contrary is established, investigators should consider the procedures that cause pain and distress in human beings may cause pain or distress in other animals. So the level of stress, all right? So you have to be in mind that animals, especially the vertebrates, so that's why when you conduct the experimentation, like here in USA and in Malaysia, if you conduct your experimentation in invertebrate like worms, insect, okay, that's all invertebrate, you don't have to apply for ethical clearance. Why you consider it only vertebrates? Because vertebrates, they experience pain like us. I do not say that invertebrate do not experience pain, but they do not. They experience pain, but different type of things. Because the scientific argument is that if you look at the pain receptor, our body system, when you induce pain, whatever, you have your neural system that is actually detect the pain and send it over to your brain. Okay, the whole pain detection is not as well established if in insect or in the invertebrate. So invertebrate, they do not have that complete system. So they argue that the way they experience pain is actually different type of us. All right. So that's why they say that we don't have to apply animal ethics clearance, because when you experience pain, how much pain that you can give to animal, then you need to consider pain in yourself. We've got like one studies. They propose to conduct experiment on rabbit skin. They want to put some hot plate. All right, they want to put some hot plate to rabbit without giving or considering giving the anesthetic agent to rabbits. And we asked them how hot is the hot plate or the hot wire, and they say, oh, that's not hot. But through our, when they submit the application, we know that it's quite hot. Then they definitely need to give the anesthetic agent to the rabbit, but they don't want to give it. Okay, so the reason is not clear, but we said that if you decided not to give it, can you please perform the experiment where that wire, the hot wire, you put into the back of your hand first. All right. If you can show us that you can stand that the pain, whatever caused by that hot wire, then you can perform on that rabbit without giving anesthetic agent. But they refuse. Of course, they say that, oh no, it's going to be very pain to human. Of course, if you pain in human, it's going to cause the same pain in rabbits. As simple as that. Okay, so after that they say, oh yeah, they don't want to give the a certain type of things because they fear, they are actually worried about whether that chemical is actually interfere with the experiment, whatever. So we just like, then we said to them, we've got like different option of drug that you can give. So they finally they consider that, all right. So that is the things that you need to consider. Okay. And then, of course, just now you see this 3R principle that I also mentioned about 3R principle. And of course, Prof. Aida this morning also mentioned about this 3R principle is a replace, reduce, and refine. Okay, if you can replace the animal, you better replace with other techniques. If you cannot replace it, you have to use the animal, you have to reduce the number of animal that you use. Okay, by calculate the the statistical or the what you call it, the sample. Okay, the number of sample that you want to use, or you can use that in combination with the alternative technique. Then you can reduce the animal, you can reduce the size of animal, the things that I just mentioned. If you have option to use rabbit or mice, just go for mice. Okay, so you apply the reduction, the principle of reduction. And refine is actually you the way you carry out the experiment to make sure that you cannot avoid, okay, no, you cannot avoid or eliminate suffering and pain. You only can reduce that, all right? Because you cannot eliminate that, all right? So, but you can reduce the suffering and by actually providing better housing and improvement to the procedures. Okay, better housing for the animal, better uh, uh, well-being or the things that you take care of the animal, the way you take care of the animal, and improvement in terms of your procedures, including that having training for the procedures. It's just like the student, when you do masters or PhD, you want to perform animal experimentation. Normally, we ask you to go for the proper training of the procedures that you are going to conduct. Okay, with the proper trainings, of course, you are going to minimize pain and suffering to the animal. Of course, this is you cannot argue with that. If you are not properly trained, it can cause a lot of problems. It's just like when you go to the doctor, you go to dentist. If they are not properly trained, are you going to give yourself to be perform surgery or whatever if you know that doctor is not properly trained, because they are going to cause a lot of problems. So the same thing here. So we want the student or researcher who want to conduct the experimentation to undergo the proper training. Okay, so make sure they are very competent, they know what to do, all right, and they are in terms of techniques, they master the techniques. So that's why that's the things that really important, because this is can minimize pain and suffering to the animal. Okay, so this is the 3R principle. So number 5, procedures with animal that may cause more than momentary or slight pain or distress should be performed with appropriate analgesic or sedation. They must be performed under sedative condition. Okay, that's why we the threshold. We have like a schedule, certain procedures, like when you want to draw blood from tail, okay, of the animals. Normally, tail snake, draw blood, you don't have to give the anesthetic agent, but you have to, if you want to do the tail snake, then you have to provide some local anesthetic. Okay, you want to do the cardiac puncture, collect the blood, then you have to give some anesthetic. And of course, surgical and other painful procedures is a must. Should not be performed on an anesthetized animal. Okay, you need that. All right. So because like here in USM, we have our animal facilities, and we have the vet officer working with us that can advise you on the performing the surgical procedures, and they are the one who's actually licensed to give the anesthetic agent. They just like, you know, in the hospital you have doctors, and some doctors is actually NS, all right, who can actually give or provide anesthetic agent. Like dentists, they are both. I mean, all the dentists have the authorization to apply anesthetic agent within the oral cavity. And just like the vet officer or vet surgeon, they also actually licensed to administer anesthetic agent to the animal. All right, so that's why you need to engage with them, because you need to know what type of anesthetic agent, how much it's going to be used, and they are going to deliver that for you. Okay. And then animal that would otherwise suffer severe or chronic pain or distress that cannot be relieved should be painlessly or humanly euthanized or killed. This is the one that when you propose your animal experimentation, we are going to ask you what is the threshold. I mean, at what stage, if your animal suffering, that you are going to terminate your subject. your experimentation. So you need to spell out when you apply for ethical clearance. Okay, we need to discuss before you are given a license to perform your experiment. So you need to know that you are aware about the situation where the animal suffering. Then you need to terminate your experiment. Normally, if your animals before your experimentation, you need to weight your animal. All right. So normally the catch is that if your animal within the experimentation lost 10% of your body weight, okay, because of your experiment, then they are suffering. So you need to consider to terminate your experiment and humanly terminate the animal as well. Okay, they are losing 10% of their body weight. How do you know? That's why you need to measure before your experiment. What is their weight? Okay, so during your experiment, like your experiment for 2 weeks, all right, then you have to weight your animal frequently to make sure that they are not losing more than 10% of their body weight. So that's the measurement normally that we use. Okay, this is a big part of that. The living condition of animals should be appropriate for their species and contribute to their health and comfort. So you need to know that different species will have different requirements. Like human, that's okay, all right, because we are about the same. Okay, for animal, no. Normally we use rats, we use mice. Even rat and mice also, their although their food is quite similar, but their caging system, of course, different. How many animals that you're going to put into one cage is also different. Okay, you need to know that. You have to avoid overcrowding of your animal. You need to provide appropriate food. Okay, enough food. All right, and then for rabbits, they have certain condition for the rabbit as well. Okay, so housing, feeding, care of all animal used for biomedical purpose must be directed by a veterinarian or other scientists trained as experienced in proper care, handling of that species. So handling of animal also is quite important. So that's why all of the students who actually involved in animal experimentation need to go through animal handling workshop or training. Okay, number one, animal handling, how to handle the animal. Number one, you handle the animal. Number one is you don't want to hurt the animal. Like rabbit. Rabbit, they are very fragile. Their backbone is quite fragile. If you handle wrongly, you're going to break their backbone. If they are struggle, they're going to break the backbone. It's quite critical. Like rodents, like the rat and mice, if you handle is not in the proper way, they are going to bite you. Okay, like the bigger animal, horse, they're going to kick you. Okay, that's why you need to know how to handle the animal. It's going to cause a lot of issues. I mean, injuries. It can cause you injuries. It can actually maybe transfer the disease from animal to you. Because if they bite you, there's going to be a major problem. So you need to know, you need to go through the process or the training of how to handle the animal. This is actually the handling, the how to handle the animal. And another one, as I mentioned, that you have to go through the training of the specific procedures that you want to perform. Okay, handling is another thing. Is then now you have to go through the specific training for the specific procedures that you want to do. So both are crucial. Then investigators and other personnel shall be appropriate, qualified, and experienced for conducting procedures. Living animal adequate regimen shall be made, training, okay, then proper use. You need to comply with all the lab animal science, care, laboratory, animal husbandry, nutrition, behavior, and everything. Okay, this also includes is what I mentioned just now. All right, and a lot of resources that you can go through, you can read, okay, and you need to go the proper training as well, and you can discuss with your supervisor. Okay, so everything need to be in place and need to be well planned. Okay, so lastly, This is the 9 principles. So the number 9, we need to have the animal ethics, or a body they are going to look at what you want to do, because normally scientists or human, all right, as I said here, human will attempt devastating and immoral practices on the vulnerable, like, of course, the small animal, they are vulnerable, to determine the outcome without ethical committee. Okay, they are very happy to do whatever they want. No, you cannot do it here, because as Prof. Aida mentioned just now, we have the act. The act is actually the governing principle that we have, the governing act that we have. If you perform cruelty, if you perform something that is not right, that is not acceptable, and considered as a cruelty to animal, you will be taken to the court. You can be charged, you can be imprisonment, it can be in you will get fined up to 200,000 ringgit for your act on animal. All right. So if you are a student, you won't get your masters. You have conduct your experiment. The experiment must be stopped, and after that, you will not get your masters. All right. You will be blacklisted as well. This is quite important for you to comply with the animal ethics regulation. Okay. So this is actually the end of my lecture. Is there any question or anything that you want to discuss? So I will be happy to, to answer your question. Okay. You can put into your chat board as well if you have something. Is there any question? Or the two students in form just now? You want to ask me question? Or you're feeling tired already? Because Prof. Aida just said to me just now, before I start my class, they said, oh, this batch of students is quite active. They actually answer all the question that she asked you. Then now how come that my question is actually, you don't want to answer my question? Or you don't want to ask any question to me? No question, that's good, because you understand what I said, right? Quite clear? I know that you are not interested in doing your animal experimentation at the moment, for your masters, but maybe when you do your PhD next time, okay, you want to actually use the animal experimentation. Okay, so if there is no question, then we can end our lecture here. What I need to know. Okay, so thank you very much.
Sampai kerja mau.